This is a posthumous impression of how Robert Hooke looked to a particular artist. So that artist has painted his painting. Next slide. This is a painting praising Robert Hooke. You can see the inscription below. Most ingenious person. Next slide. This is a picture, self-drawn picture of Robert Hooke's microscope. You can see the eyepiece, the swivel, the stage, the lighting, how the light is getting focused. Though heavy and primitive, it is really ingenious. Next slide. This is the method by which Robert Hooke constructed his microscope. Along with it, he has given all the details in his book, Micrographia. Next slide. This is his famous picture of cells observed in cork. The cork he has used from the branch which is drawn below. As you can see, the tiny hexagonal honeycomb shaped rooms. Next slide. This is about how King Charles II funded his experiments, and how the technology used was microscope, and his association with Royal Society of London. Robert Hooke was a fellow at Royal Society of London. This was January 1st, 1653. So King Charles II had everything funded for Robert Hooke. Before him there was a person, and then next Robert Hooke came and continued this work with the microscope. He was a self-educated child and a child prodigy. So to say, he was a genius. Next slide. This is the first page of Micrographia. You can read it. Next slide. His letter to the king in the book, thanking the king for all the funding. Letter to the king continues. And finally, letter to the king ends with him saying, "Your Majesty, humble and obedient servant and subject." Next is his letter to the Royal Society of London, praising his colleagues who helped him and supported him. He observed many things other than cork. So he observed things at the end of the needle, like this. Things at the end of the needle he observed under his microscope. He observed silk fibers. You can see silk fibers, the patterns formed. He observed flint. By striking flint you can create fire, right? He observed flint. He observed frozen objects, frozen in water, frozen objects. Yes, frozen objects, as you can see. He observed charcoal and charred vegetables. See, charcoal, charred vegetables. Then this is his picture again of cells of a cork. This is observation of a saprophyte like mushrooms, etc. Spores basically. Observed bramble leaves and other different types of leaves he has observed. Seaweeds, he observed different types of seaweeds. These are the patterns observed in the seaweeds, as you can see. Then he observed poisonous plants like nettles. See, you can see the sharp pointed hooks of nettles, poisonous plants. He observed seeds. You can see different types of seeds which he observed. He observed skin, he observed hair, as you can see. He even observed flies, house flies. You can see the texture of the housefly, the big drawing, the eyes of the housefly, as you can see. He observed the teeth of snail, as you can see. Teeth of snail. He observed the teeth of snail. He observed gnats or water insects. You can look at this water insect here. He observed it under his own microscope. Then finish. Observe the spelling of finish. It doesn't have an H. It's Old English. And then he's written the table of contents. Thus we know that Robert Hooke was the first to discover the boundaries of the cell. That is the cell wall. That is how he could say it is like a small room or honeycomb like structure. Thank you, friends.